almost impossible. To really work on forces and work on proteins is not so easy. But we work on genes because we know how to do it. But actually, we would like to modify proteins. And let me illustrate you with this movie here. This is a Drosophila embryo. This is the anterior end. It's viewed from the top. So this is the extended germ band. So the embryo starts here and then goes along this way, and then the end comes here. So the end of the embryo is here. So the germ band is fully extended, but that's you'll see how this embryo does now. It's a movie that lasts. It shows germ band extension and dorsal closure. So these are developmental biology things. This is not a gene X coding for this. So this is what happens in the development of a Drosophila embryo. So the germ band goes back and you have dorsal closure occurring. Again, I run it. So this is a sort of a very fast movie of quite some time, but you see a lot of things happen. And the nice thing about movies, you can go backwards. So let's start when the embryo is made and then ask, what do you see? So here suddenly the end opens up and then the germ band goes back. So let's run it this way. You can go now back and forth and look at it again and again. And probably some of you are looking here, some you look here, some look here, some look here. I mean, a simple movie like this is a source of millions of things. So look here about how dorsal closure occurs. And now, after a few times, it doesn't want to play anymore. So this is the dorsal hole that closes here. You can also look here at the trachea as the movie runs. Here in the back, and then you see how the trachea forms here. You can look at the head. So there are millions of things that happen. And a lot of these things, okay, the movie summarizes maybe 4 or 5 hours of Drosophila development, but within each of the minutes, things happen, cells move, cells pull, cells through. So it's not a 4-hour effect. It's a second by second or minute by minute, proteins doing their job, working, pulling, pushing, dividing, changing their shape. This gives rise to this particular embryo at the end, with all of these ongoing business. Now you can imagine if you make a mutation in a gene, all of these cells are mutant. So you don't have a control. You don't see much. If it's an important protein for forces, then you don't see anything. Nothing moves anymore. If you make a mutation inducible in a certain group of cells, but until you make it inducible, you still have the wild type protein out there. It takes half an hour to try transcribe the gene, then you have to have the RNA, you have to splice it, export it, make the protein. The wild type protein is still there, so you have to wait until the wild type protein is gone and the mutant protein takes over its job. So genetics modify the genome to ask what a protein does in this living embryo here is not possible. It's not the right way to do it because it takes far too much time. And developments happens. The cell activities, they happen at the second to minute range and not at the hour range, especially with regard to morphogenesis. So how do we deal with this? We would like to modify proteins acutely in specific places. As I said, the cell is the business end. And the cell is made up of proteins and lipids and not DNA. That's the nucleus. So how do we go about it? There is a real problem for the genetic approach. I mean, we are on the one hand so spoiled with having CRISPR to do genetics, but at the other end, we do know that what we can do is very limited with regard to what we are interested in the sense of understanding what proteins do in this dynamical fashion, morphogenesis fashion, how to organize things, how cells divide and so on. We can't do much. Trafficking components, they're needed everywhere. Actin cytoskeleton is needed. Actomyosin is needed. The Drosophila embryo is loaded with maternal proteins because it has the same size as the larvae. And this maternal embryo, the maternal egg, is full of proteins. If we wouldn't have any proteins, we couldn't develop this embryo, which also means that for most of the genes, we cannot get rid of the protein by having a zygotic mutant. The egg is already full. In order to make an egg, you need a mother. You cannot remove proteins there. So again, genetics will allow you to look at the subfraction of the function of genes. And these are the genes that are required in certain tissues. They make the protein only in certain tissue. So of course, you can only make a certain protein in a certain tissue once this tissue is made. But RAPS actomyosin is needed in all the cells, so the mother puts it in the egg. That's what you get from your mother, and you go with it. So you cannot do genetics on this because the mother needs it as well. So there are loads of problems to do developmental biology in this live manner, because we would like to acutely change proteins. Go like this: protein X, remove it only in this cell. What is the effect? We cannot do that by genetics, only for certain proteins, and it takes 2 or 3 hours. And by that job, we don't any know what are primary effects and secondary effects. So what would be the solution? The solution would be to deplete proteins or to play around with proteins in the cytoplasm or secreted proteins in a very specific, targeted manner. You make a movie, you see how something's developed. Then you make a movie and you remove protein X in a certain cell, or you put it to another place, and you let the movie play and see second by second, minute by minute, hour by hour, what the consequences are. That's not what we do with the mutation. We delete the protein, we wait until it's completely gone, usually, because we delete it everywhere, and then the bad thing is done. The protein is gone. So we only see what happens at the very, very end of the process, not really what happens during. Of course, since we can use live imaging to document development, this urge to do things with proteins and do it acutely has become much more obvious, because we see in a movie how A develops to B to C to E. Before, we just took stills and saw, okay, if I remove this, no I. But why no I? In a live movie, you can see it, and then the questions become much more defined.
Looks like we're here till May 11th. You're on mute. Mm-hmm. Looks like, because I asked the communications office what's happening.
Fraser.
And they just answered. Said, don't share, but
Fraser.
May 11th.
Alessandra.
Yeah?
You have to turn off Fraser. Say hello to him and mute your microscope. Sorry. Okay, so the solution to this would be to delete proteins, very specifically, maybe even, and this is very interesting, once the organism is made and you want to ask whether a protein is required for memory, but you can only study this if you first take a person that is, or a fly that has a memory, and you train it for a memory, and then you delete the protein specifically and ask, is this protein required for memory formation? All kinds of questions are absolutely need the protein depletion methods. But this doesn't really exist, or this didn't really exist until a few years ago. There were some things developed. There was a protease developed that doesn't exist in flies and in other organisms. And the protease cleavage site, you could put it into your gene of interest. Then the protein has a cleavage site once it's made. And if you express your TEF protease in a certain tissue, the protein will be cut only in this particular tissue. That was published in 2008. I loved this method when it was published by Kim Naismith. We tried to do it in flies, but it didn't really work because we didn't have CRISPR to insert the protease site, so it was very complicated to do it. And then in many cases, the protein doesn't cut the site because it's not accessible. So the method, it was one of the first ones to do induced controlled protein degradation, but it hasn't been used a lot because we neither had the tools, nor it is an elegant method that works in many ways. And you don't degrade the protein, you cut it into two. So often the protein in a big complex, you can cut it here or there, it stays in the complex and functions. So it was the first initiative by people to try to manipulate proteins. In 2008, there were systems like degrons, where you could add sequences to a protein, and these sequences targeted this particular protein to the proteasome, that in bacteria and in yeast. So there was some things were around, but actually quite little. Nothing in flies beside the TEF system, which didn't then work for us. But it definitely was the first sort of glimpse that maybe we should work with proteins, and acutely post-mitotically. Post-mitotically, you cannot really do genetics. You don't get clones, you don't have cell division anymore, and so on. So we want to manipulate protein. We need to manipulate protein. We need to manipulate DNA with CRISPR, that's clear. But in order to get more insight into cell biology, we need to modify proteins acutely. I think that's clear now, that this is what we need. The problem is, as I said, many proteins are stable. If you do genetics, you don't get rid of them. So you have to acutely deplete them, and so on. The solution would be if we would work with proteins, if we could recognize proteins. So that Emmanuel Cosinus, he was a postdoc in my lab from 2006 to 2013, maybe. He's now in Zurich. He made a lot of movies of tracheal development and how cells divide, or how cells migrate and make branches and so on. And we wanted to ask, what are the forces that transform the tracheal branches? You will see more about how branch networks are made tomorrow in the vascular system. But anyhow, he asked that question because he wanted to remove forces. Forces are made by actomyosin. But of course, if you deplete actomyosin, the cells don't divide, then you don't get a tracheal system. So genetics was not really possible. The embryo is loaded, the egg is loaded with actomyosin. So even if you have a mutant, the protein is still there. So there was just no way. You can use chemicals, but then you inhibit actomyosin in all cells, and then you don't even know anymore what is cause and consequence. So he was in Heidelberg at that time, working on laser ablation with a colleague on the tracheal system, but he was in Heidelberg, and then he apparently thought about this problem. He only came to tell me about this new method once it was working. I didn't hear much about it, so I don't want to take credit for it. But I can imagine that what he thought is, how can we work with proteins? We need to degrade, let's say, actomyosin acutely once the trachea or an organ starts to form, and then see what happens. Can cells not move? Do they need actomyosin to move, or do they not need it? Tracheal cells don't divide once they're made tracheal cells. They make the three-dimensional structure without cell division. So we could ask where the forces come from. But there was no genetic approach to it. So how do biologists recognize proteins? You know, how did we recognize labial? In this mess of the whole Drosophila embryo, we used an antibody. And we coupled this antibody to a secondary antibody to make it visible. But we first had to open up the embryo, of course, and kill the embryo and permeabilize the tissue and fix the tissue. And then we could add the antibody. The antibody would go into the nucleus because it was permeable. And then we can see where the protein is. Antibodies are used in every biological study of the world, in millions of papers every year. It's the CRISPR kind of thing of proteins. An antibody recognizes a protein very specifically, like RNA. It's very, very specific because the antibodies have been developed by the organism to fight non-self proteins. If they wouldn't be specific, you would auto-digest your body. So by definition, they're very specific. Otherwise, you would have a real problem. So we use antibodies. Antibodies are big things. There are two chains, usually a heavy chain and a light chain. There are a lot of cysteine bridges that hold the heavy chains together, and the heavy and the light chains. And the antigen recognition region is up here. And as I said, they're very specific. There are hundreds, thousands of monoclonal antibodies that have been isolated. It's a tool now also in the clinic, because antibodies, the new cancer drugs are all antibodies. So it's used to recognize proteins, of course, only in the extracellular space, because this antibody cannot work in the cellular environment. Actually, if you force it and inject it into a cell in culture, it will stay folded kind of and recognize its protein, but you have to inject it. People start now and go back to antibodies to do cell biological studies by injection. Not very elegant, but it can be done. Now, a chance discovery, it's like with CRISPR and talents. These were discoveries made in funny conditions. This is actually very interesting because Hammers, a guy from Belgium, he was an immunologist and he had a lab like we do now. He guided a lab and people were looking at antibodies in different blood preparations of mice and camels and who knows what. So he apparently, the story goes that Hammers told a student to go and fetch the mouse serum in the fridge and then run a gel with it. And then they would analyze together these different species, the heavy chain and the light chain. And so the student could see that antibodies are made of heavy and light chains. So the student apparently went to the fridge, took the sample, put it on the gel, and there was only one band. There was only a heavy chain. So of course, what you do first is blame the student. What did you do? I mean, what did you load here? I mean, it can't be an antibody has two chains. I'm sorry. So the student, I guess, had to go back and try to find the sample again that he took. It turned out that he didn't take the mouse sample, but he took the camel sample. I don't know why he had camels in this sample, but Muldermans, he's a guy from Lebanon, and he was his student. So maybe Muldermans, and he's very famous now, Muldermans, because he co-discovered these single-chain antibodies. He might have brought these camels somewhere from Lebanon. Anyhow, it turned out that camels have normal antibodies like everybody else, but they also have these single-chain antibodies, published in 1993. First, only organism. Later, sharks. Some sharks were shown to also have these antibodies. We only have these here. But camels, for a reason we don't know, they have antibodies like this that make only a heavy chain. It is due to a mutation that does not allow the light chain to be, what is it again? It's a splicing mutation that results in this thing not being able to recruit a light chain. But probably through mutations, these heavy chain, the regions that recognize the antigen has mutated in a way that it has longer side chains and it recognizes the antigen with the same high specificity, also it's only one chain, as normal antibodies. Now this region here, you can clone it. It's 100 amino acids. So it's much, much smaller than this, of course. This is a dimer and this doesn't come alone. You have to build a heavy light chain stuff. So with these 100 amino acids here, you can fold a full antibody with its full repertoire, its full binding specificity. You can clone this and express it in a cell. And in contrast to this works in a cell. So suddenly people realized, whoa, we have here antibodies, soluble antibodies that we can express as very small moieties in cells. They bind to the protein in cells, and now I can recognize any protein I want as long as I have an antibody against it. And if you want one, you inject your protein into camels and isolate the antibody. So suddenly, in 93, the idea came up that, oh, now we can recognize any protein we want, and we can even recognize it intracellularly, and maybe we can do all kinds of with these proteins, because now we have, like with CRISPR, we can target the protein specifically. CRISPR targets a DNA sequence. You decide what you do at this DNA site. You make an insertion, a deletion, a reversion, and so on. CRISPR only recognizes and cuts the site. Here we have suddenly now a tool that would allow us to recognize proteins, and then it's your imagination of what you want to do with it. But you can go and recognize and find your protein in the cell at the time you want, maybe. So these were ideas that came up. because these things function intracellularly. Of course, in flies, we can express as a transgene anything we want, and they can express it anywhere where we want. In pharmacy, you can't do that. So this thing, as you can say, it's called Abling's nanobody, was immediately patented because you say, if you manufacture antibodies for pharmaceuticals, this is much simpler, cheaper, better. You can scale it up and so on. So they immediately founded a company and called it Abling's, the company. This thing was patented, was called nanobody, so you couldn't really use it for many other things. And the company then also prohibited a lot of people from really doing much other work. Often they couldn't publish. So for 20 years, I would say 93 to 2003, not much happened. And after slowly, things came up a bit. But the ideas were kind of there in these days. We'll come to what happened with Abling's. So we have like a CRISPR type of thing. We can recognize proteins within the cell. So maybe one could use this. Of course, there are no nanobodies at that time when Emmanuel started to work on it. Almost up to now, there are no nanobodies against fly proteins because the fly community doesn't work with nanobodies. They work with CRISPR. They don't work with nanobodies. In 2008, of course, nobody worked on nanobodies beside the few groups who wanted to make therapeutics. And one of the guys, Mildermans, in the early days, he had a guy called Heinrich Leonhard. He said, nanobodies, this is fantastic. We can do a lot of things. But then they said there is no nanobody against anything that is to interest of anybody besides pharmaceutical. And he said, in 93, it was published. I think in 95 or 6, I met him recently. He told me. In 96, he said, okay, wow, why don't we make a nanobody against GFP? Suddenly, all the labs started to work with GFP. He said, maybe it would be interesting to have a nanobody against a protein that everybody works with, because that might allow us to do a lot of things. So this antibody the injection was done in 96, I think, 97. The antibody was published only 10 years later, 2006 or so. This is the structure of this nanobody. You can see it's very small. It's smaller than GFP. We have introduced, many labs have introduced GFP into many genes to make fusion proteins that are fluorescently labeled. You see, if we can insert GFP, we can certainly make a lot of things with nanobodies. They're even smaller. This interaction is very specific. The nanobody doesn't recognize anything in the fly unless this fly expresses GFP somewhere, and then it will recognize GFP. So there was this nanobody available from the Rothbauer lab, and Heinrich Leonhard actually had the idea and did it. So the idea of Emmanuel was then, okay, if I want to manipulate proteins, maybe what I could do is I add the GFP to my protein of interest. This we do anyhow nowadays. As soon as we want to work with a protein, we try to label it with a fluorescent protein so we can see it. And in a live move, we can see where it is, where it goes. Is it diluted through cell division? Is it made again? Where does it go? How does it work? So we usually use GFP anyhow. But if we have now an antibody, a nanobody that recognizes GFP, we could grab this fluorescent protein. And then maybe here is the mind is the limitation. Here you can think about what you want to do with it. Emmanuel wanted to degrade a protein, and he wanted to degrade actomyosin. So myosin regulatory light chain would make actomyosin inactive. So how could he do that? He could put an F box to this nanobody, and this you do in a transgene. So you make a transgene on a P element that expresses this nanobody, maybe in an inducible fashion with Gal4, maybe in another fashion. But the nanobody, this coding sequence of this nanobody is fused to a so-called F box. And an F box is an amino acid sequence that drags the protein to the proteasome. So if the nanobody F-box construct binds to the GFP, then this GFP fusion protein will be brought to the proteasome and be degraded. That was his idea. F-boxes were known. They haven't been used in flies for any manipulations. Nanobody was known. They haven't been used by anybody in the fly field to do anything. So that was a brave idea to do that. So he tested it. So this is the idea. Usually proteins bring substrates to the proteasome to degrade them. So the substrate binds here via this domain and then is degraded. So what Emmanuel did, he used as a substrate GFP, and he used as this domain that recognizes the substrate here. He used the nanobody. So the nanobody would recognize GFP, and then the GFP protein would be degraded. And he tested it in cell culture. He took a histone protein, histone 2b. This goes into the nucleosome. It's in the nucleus. So I would never have tested histone 2b to be degradable because I would have thought this is embedded in the nucleus, in nucleosome. So it's very shed away. Is there a proteasome in the nucleus? I don't know. But he used H2B, and this is the one figure of his paper in 2011, 2012. This is all the nuclei are green in this cell culture experiment. But in the transfected cells in A here, he added the nanobody that recognizes GFP fused to an F-box. So nanobody F-box. This is F-box, this is nanobody. And then the green is gone. So a cell that expresses the nanobody that binds to GFP with an F-box seems to put this GFP protein to the proteasome and degrade it, and now it's gone. If you have only the nanobody or only the F-box, it doesn't work. So you need this combination of the nanobody with the F-box that grabs the protein and brings it to the proteasome. So this seemed to be working very well. Of course, this was an experiment that takes 2 days. You do transfection, and then you split the cells and you feed them, and then you transfect them, and then you wait 2 days, and then you look at it, and then the protein was gone. What he did afterwards, and this is a fabulous experiment, he asked, does that work in flies? Because nobody ever had used nanobodies in flies, and the F-boxes and degraded fluorescent proteins. So, and he was a perfectionist, so he made a fly that has at the same time histone 2A expressed in all the cells with a green fluorescent protein, fused to a green fluorescent protein, and the histone 2A fused to a red fluorescent protein. So all the cells of the embryo are yellow. They have a green fluorescent H2A, and a red fluorescent H2A. Now, if this nanobody who binds to the green protein with this F-box should only degrade the green protein. And if you express this nanobody in stripes, you should remove green from every second stripe, but the red is still there because it should not be degraded. The nanobody doesn't recognize this red fluorescent protein. So you should generate from yellow cells, you should generate red stripes because you remove in stripes the green protein. So look at this movie here. All cells are yellow to start with. The question is, does the nanobody degrade the green protein in stripes? And we use the stripe enhancer to drive this nanobody F-box construct. And this is the result, germ band extension, coming back. So obviously, the nanobody degraded the protein in these stripes here. So this is all the nuclei in a certain field. All are red. These are the same nuclei, exactly the same nuclei, but looked at the green protein, and the green protein is gone in these nuclei. Here, it's gone. Here, it's gone in the second stripe. So definitely you can degrade proteins in flies in an inducible designed manner. Here you want to degrade the histone 2A that is fused to GFP, and you do it. And from this movie here, you can, of course, go back and forth. And that's, again, the nice thing about movie. You can say, how long does it take to degrade the protein? When is the stripe enhancer induced? So you go back, you have stripes here. So this is germ band extension. At that time, a little bit earlier, the degron is made. The stripes express this nanobody fusion construct. And then you go 10 minutes later, 10 minutes later, 10 minutes later, 10 minutes later. Oh. So it takes about half an hour to degrade the green protein. Do the cells divide? Do the cells need to release histones and divide and then rebuild it? No, they don't. You can look at, there are millions of things you can look at in this movie to show how specific the thing is. Does it require cell division? How long does it take? And it turns out it goes very fast. So you can induce and degrade your protein of interest. So that's now nice, because now we can degrade any protein we want on a piece of paper, because we fuse GFP to it, and then we have a fly which has all our protein of interest labeled with green. We can make a movie. Is it a nuclear protein? Where is it made? Where does it distribute in the cell? Already loads of information on this protein just by looking it in the wild type. And then you can ask, okay, now I understand where this protein goes. What does it do then? So you can remove it from a few cells by expressing this degron in this specific cell at the time you want, by using enhancers that come up. Then you can probably make it light inducible and so on. So here we go. We can think about doing a lot of things. Most of these things have not been done. This was published in 2012. So the proof of principle: have a nanobody that is fused to an F box, and this nanobody recognizing a protein, and then you can degrade it. Of course, you can now have your nanobody going against GFP. Then you degrade GFP fusion proteins. You can isolate nanobodies that go directly against your protein by injecting the protein in camels, and then you can go to a wild type embryo and degrade your proteins. And. The sky is the limit. But first you have an antibody against your protein of interest. I'll come to that. This is now all feasible. There were no platforms to isolate an antibody in 2010, 2011. Now every company sells nanobodies. You give them your protein of interest, the sequence of it, they clone it into yeast, they isolate your nanobody, they send it back to you, and then you have it. And then you can start. All of this was not existed when Emmanuel did these experiments. He had this GFP nanobody, proof of concept. But what we wanted to do then is address biological questions. Nice method is good, but can we really address biological questions? And the biological model we looked at is this, dorsal closure. This system has been used by a lot of labs, not by ours. We've worked very early on dorsal closure because thick vein mutants, they fail to close up the dorsal hole. So DPP is involved in dorsal closure. We still don't know the details about it, but it's involved. So it's still early days. But this is the process we wanted to look at. How does this dorsal hole here close? And there was, in the published literature, there were 3 forces that are involved in this. It was the amnioserosa cells here, these cells in the middle. These are very big cells that disappear. They are apoptosed later on. They develop forces that pull. There is an actin-based actin string here in these epithelial cells here that is supposed to be required as well. And there are zipping forces here. There are 3 forces that are proposed to be involved in dorsal closure. All of them needed to close up the dorsal hole. So there is a blue force, a red force, and a pink force. And that was the model based on all kinds of chemical experiment and genetic experiment. But now, if we could ask of this, all of these are actomyosin-based forces. So if we would express the nanobody only in this tissue here, we could ask, is force or actomyosin-based forces required in this tissue? Does dorsal closure still occur if I only inactivate it here or only inactivate it here? So we immediately realized that we could attack these problems, but we were not working on dorsal closure. So we joined forces with Damian Brunner. Some people say he's my brother, but he's not. If he would be my younger brother, of course. This is a older picture even of him. So what Damian published in Cell is that the amnioserosa cells, these big cells over the embryo, that's how they behave. They pulse. This is an actin movie, and you can see the cells pulse. And every time before they pulse, they synthesize actin. They put myosin on it, and then they contract. So he was the first to describe. And here, this is a movie which lasts about, the contractions are about 1 every 12 minutes or 16 minutes. So we talk about minutes. If you want to understand what happens here with this dynamic, you cannot use genetics because it takes 2 hours to deplete the gene. Once you mutate it in the genome or make a clone, and the endogenous protein is still there, then you introduce the mutant and you have to deplete the wild type protein. It's just not doable with genetics. So we asked Damian whether we could collaborate and ask whether actomyosin, you can see these actomyosin forces. You see actin, and then myosin goes on it and the cell shrinks. Actomyosin is pulling forces that so make the cells shorter. So of course he hypothesized that these forces are important, but he also hypothesized that they need to work in conjunction with another, with this pearl string out here. So the Cell paper that he published in 2009 said that these amnioserosa cells here, they generate pulling forces, and apical surface oscillation, and these forces, together with the actin string, which is not shown here, generate dorsal closure. So the cells pulse, and each time they're smaller, the ring around it sort of pulls it in. So the cells they become small, they do not extend to the initial size, and then they pulse, and in this way they get shorter and shorter because you kind of, you pull the string. It's like a ski boot. You go in the first thing and then in the second, and at the end it's really tight. That was his model in 2009. And we said we can maybe look at it now. Is it really the pink forces and the red forces that are required? So what we did is we expressed this degrad only in the amnioserosa cells, these pulsing cells. And this is a movie, you might not see it. The cells don't pulse anymore. The cells still live, but they're dead. They don't generate any, this is Brownsian motion, what you see now, more or less. The cells don't pulse anymore. They don't have these actomyosin dependent forces anymore. This is the cable that is proposed to be important as well. The cable is normal because we only degraded actomyosin in these cells. So we had a fly that had the myosin light chain fused to GFP. Squash is the myosin light chain. We had a mutant of squash, and this mutant was rescued with a transgene that had squash GFP. So the only protein that can make actomyosin here, active actomyosin, is the GFP protein. And when we deplete it only in these cells by expressing degrad in the amnioserosa, we only deplete forces in the amnioserosa. All the other forces are normal. The question is, what is the phenotype of that? The forces are inactivated is shown in this movie. There's no pulsing anymore. I mean, in contrast to what you saw before, these nice movements, they're gone here. So first of all, the method works really well. And then the question is, what do we find biologically? And this is the answer. So if we delete actomyosin, we inactivate pulsing forces in the amnioserosa. I've shown you before. We've actually shown it in our initial paper, so we knew kind of the result. This is what happens in wild type. Dorsal closure occurs. And this is what happens in the mutant. Dorsal closure doesn't occur. Dorsal embryo spills up. It's exactly the thick vein phenotype too. Without actomyosin, you don't close. Without thick vein, you don't close. But we still don't know what thick vein does. So that remains to be looked at. But definitely, forces are required in the amnioserosa, and if you don't allow them to develop, then dorsal closure doesn't occur. So this is clear. But Damian proposed that if you inactivate the string forces around it, so pulsing important, the string important for have the pulse get smaller and smaller, he said there are 2 forces that are needed. So what about the other forces? So what we did is to deplete actomyosin only in the red cells here, in the epithelial cells, and leave the pulsing normal. So the question is now the pulsing, if you don't restrict the pulsing, then you could say, now they also they pulse, but they don't close because you don't pull it in. So what is the phenotype if you only inactivate actomyosin here? If you inactivate pink, no dorsal closure. So you need this pulsing. But do you need pulse and string here or not? So we inactivate it only here, pulsing occurs. What is the phenotype? So this is the phenotype. This is the normal closure up here. And now if we go back to this movie here, you can see that dorsal closure. What is this? Oh, this is, oh, actually it's the wrong movie. I have the other one here, so it's good. I thought it would close, but I can see myself from a movie that it doesn't. This is again the movie with amnios serosa, the pulsing cells. I showed our own movie before. This is now the movie from Damian. So you see the pore spring develops, but the amnios serosa doesn't close. So if we go back, here you see dorsal closure is the wild type. You see the purse string here. Here you see the purse string is properly made, but the amnios serosa doesn't pulse. And if it doesn't pulse, it cannot close. Here it's already closed. Up there and down it's not closed. Okay, amnios serosa is required. What about the dorsal ectoderm, these pulling forces? Here you have the movie. Wild type closes again. And the mutant closes as well. So if we have pulsing forces, but no string, no purse string, dorsal closure occurs. This is the result. So here, the cells, you don't see it, but they still pulse. But you see the purse string is gone. Here you have the purse string. Here it's gone. It's squashes degrade in the epidermis. You get these big blobs. Don't tell you why, but you see it closes, it closes, it closes, gets smaller, smaller. Like here, almost like here. So it closes. At the end, the embryo is dorsally closed. So Damian's cell published model that you need two forces is wrong. So we published this together with Damian's lab in 2016. This, of course, showed that pulsed forces timed by a ratchet-type mechanism drive directed tissue movement during dorsal closure, published in Cell 2009, without using protein manipulation methods. Now, if you use protein manipulation method with this help of what we call degrad, degradation of GFP fusion proteins, when you use this degron, binds to actomyosin, to the activator of actomyosin, degrades it, you can now say amnios serosa cell constriction, but not epidermal actin cable tension, autonomously drives dorsal closure. So we have a new method, completely new. We can degrade proteins in tissue-specific manner, in an inducible manner if you induce the nanobody. We can do experiments we cannot do with CRISPR by DNA manipulation, because we now target directly the protein and remove it. Squash is maternally provided. You can remove it by genetics. So we can modify models by using new techniques and really get insight into the biology of a developing Drosophila embryo. So what Emmanuel did is he developed a synthetic degron. So this is now, I find it incredibly exciting because this is synthetic biology. We use nanobodies, we use protein binders, and suddenly we can generate completely new tools that do not target the genome, but that target the proteome. And to me, the protein is anyhow what you want to understand. We only use mutations in genes because they affect the protein, and we want to learn more about the protein. So why wouldn't you go directly to the protein? Why? Because we didn't know how to do it. Nobody really knew how to manipulate proteins because antibodies did not work or do not work in the cell cytoplasm. But the development of nanobodies, this chance discovery by Hammers, by the student who made an error. Sometimes it's good to make errors, you see, if you learn from them. So he discovered out of the blue that there are tools like nano antibodies that can be used in vivo, and then Emmanuel developed this first synthetic Degron that can be used in flies. There are hundreds, thousands of GFP fusion flies in the stock center. You can use that for memory studies, you can use it for developmental studies, and it's all at the very beginning. But we have a genetically encoded, so we use genes, but it's a functionalized protein binder allows to manipulate the protein in the cellular environment in vivo. So this is, of course, very exciting because we can do a lot of things now. So I will unfortunately have to stop here, but let me just have a wrap up. This synthetic Degron, as you can see, was published in 2012. It was sort of the first designer-based protein manipulation system in flies. So now you have millions of possibilities, because nowadays you can have a protein binder, an antibody. Let me see, maybe I can go on another. We'll finish in 2 minutes. And you'll finish at 12 for sure, because Shinya will. So maybe I have something more here. Okay. Okay, so Emmanuel developed this Degron, a nanobody against GFP fused with an F-box. Then you can degrade proteins. We use that extensively in the lab and in collaboration. Many people use it now. Of course, you can use it also for a lot of other things. And Shinya will tell you how you can use this method now to ask more about DBP dispersion. How does DBP really need to be secreted from a cell and go away from this cell? How far can it go? How far should it go? We have no clue, because we cannot address this genetically. But targeting DBP directly, maybe trapping it, we could maybe ask some question. And Shinya, he will talk about this in the next lecture. Now, this is all done with an antibody against GFP. And of course, we would like to have nanobodies against the 20,000 human proteins or 25,000 human proteins. And this has all been developed in the meantime. There are huge platforms. You can send your protein sequence to a company, and they will, you don't even have to do anything. You just send them the sequence. You don't have to purify the protein. They will clone it in US2 hybrid and make a 2 hybrid screen with a nanobody library, and then they find your nanobody and they send it to you. They send you back 10 sequences. These are the 10 binders to your protein. Go for it. You can use it in developmental biology. You can use it in synthetic biology, of course, in therapeutics. And there are many, many. Just want to show this side. This is what a nanobody looks like. It comes from these heavy chains of camels. If you want to target a therapeutically important protein, you go now with a nanobody. That's easy, cheap. Then you have in Zurich, you have a guy who has worked on, he's an immunologist, but he said maybe we can make a synthetic scaffold and design the scaffold so that you can easily solve the structure. It's soluble, it's stable, and so on. And then we modify the residues in large libraries and screen for library members that bind to protein X, Y, Z. So it's a scaffold. It's called designed anchoring repeat proteins. There are libraries of 10 to the 11 expressing variants of this, and you can screen these libraries in an automated fashion. It's all robotics now, and then you pop up with 30, 40 binders. Again, Shinya will talk a little bit about that. So this is all synthetic. It's all roboterized. There are repibodies, there are DARPins, there are anticalins, there are monobodies, there are affibodies. Each of them has a company. Ablings has been bought now by Sanofi for 1.2 billion to generate nanobodies against any interesting things. Molecular Partner is the company for the design anchoring repeats, and the sky is the limit. You find a binder against your protein, against phosphorylation site of your protein. Against even nucleic acids, you can find proteins. And of course, maybe one last word, we have a big problem in the world now, and this is coronavirus. So I would have expected 4 weeks ago that the first therapeutical tools developed against coronavirus should come either from nanobodies, DARPins, REPE bodies, AFI bodies, because these are synthetic libraries. It takes 4 days to look for binders. So 4 days after the coronavirus spike proteins have been crystallized or structurally solved, you should have these binders now. And of course, I followed the literature on a daily basis. But last Sunday, the first paper reporting 70 or 80 or I don't know how many binders, 32 were characterized then, I think, binders against the coronavirus spike proteins were published. So you can guess, was it the DARPin scaffold that was used, or was it the anti-kaolin, monobodies, affibodies, or nanobodies? It was a subversion of the nanobodies, the first paper published on Sunday. It's a group in Zurich who had made synthetic nanobody libraries. They're called Psi bodies, completely synthetic. There's no camel involved anymore. This is nice for animal research too. So it's a synthetic library. They screened this synthetic library. They have 32 different binders against the spike proteins. Was published in bioarchives. I can probably upload the paper. So it was clear that this toolbox is going to be the future of a lot of therapeutics, but the future of developmental biology too, because we can do now everything we want. But we are at the level of restriction enzymes at the moment, kind of, because we haven't even started to do everything that has crossed our mind. But I was sure that this scaffold would be the first one to have high affinity binders and published in a quick way, and that happened now. So I just wait for the day the DARPins are published against the coronavirus spike proteins. And then the REPE bodies, affibodies, monobodies, anti-kaolins, all of these companies, they know now what they have to do, and they do it because it's 4 days of work. And these scaffolds are so easy to label. Every student can isolate 20 nanobodies in a night because they're optimized to be highly produced, highly stable. You can have cloned the tag to it, you purify it overnight, the next day you have milligrams of this binder. And what they try to do now is they couple it to surfaces, microparticles that they could inject into the lung and fight the virus in the lung. That's by inhaling. So lots of possibilities for clinics, lots of possibilities for developmental biology, because now we can manipulate proteins, the workers, the guys who do the job, and we don't have to go through the DNA to do it. So this is a revolution, there's no doubt, and developmental biology, and we profit terribly from this revolution, and of course we hope to contribute a lot to it. So now we can make a 15-minute break, and Ginio will then start, and he will start at 11 o'clock. It's difficult to say. I hope you're still there. If you have questions,